I'm Jonathan Tales. This is Microclimate and Vegetation Communities in Forest Remnants. On this photo here, we have a picture of a forest remnant that's left in a clear cut, and this is left here to mitigate some of the negative impacts of clear cutting. It's intended to emulate the natural version of this would be a fire skip found in a wildfire. So there's some hypotheses about why these might be important. They can act as stepping stones for the movement of animals through the disturbance. So animal that might be vulnerable to predation, for example, in the disturbance might find refuge here. They're going to contribute to forest structure variation over time, both initially after the disturbance and as that disturbed area recovers. They're going to provide refugia and seed sources for interior forest species. That's kind of something that we are adjacent to in this discussion about microclimate. If these have cool microclimates, then we think that they can potentially house species that may not be able to survive in the adjacent disturbance. And if any of the above are true, we expect these to have high diversity and high ecological value. And that would support their continued use in clear cuts as fire escape analogs. So here's our study. We selected several fires and several harvests in the upper foothills region of Alberta. This is an area that has a lot of lodgepole pine. That is the main species that's targeted for timber harvesting in the region. We laid out a few transects. We put a transect across an island area and then an adjacent reference site to try and quantify edge effects in those areas, which we expect to have an influence on microclimate. You can see in the bottom right corner of the slide a example of our plot layout. Basically, we had a large plot every 15 meters with some vegetation, sapling subplots. We also collected coarse woody debris, and we put these microclimate sensors in there throughout the course of the growing season. So I started by doing some data exploration. On the left here, we see a PCA showing forest structure variables in purple and our blue microclimate variables as the response variables. And we can see that a lot of those microclimate variables are aligned along component 1. And some of our predictor variables, edge distance, live tree density, and percent canopy closure, organic layer depth, are also aligned on that component. So we expect those to be correlated. On the component 2 axis, we have a bunch of things, but they don't appear to probably influence those temperature variables very much. Maybe soil moisture has something going on, but I don't put a lot of stock in those soil moisture values at the moment. On the right here, I did an NMDS of our forest microclimate variables, which are in black, those black vectors, and then our vegetation species. These are about the top, 15 or so species according to the NMDS. And then these ellipses represent our different sites. So the important thing to take away here is this pink site at the top. This is our one of our fire sites. We actually only had 4 transects for our fires, but we had 8 transects for our harvests. That was due to some access issues in the early season. And this one this one fire site is sort of a unique ecosystem relative to the others. It's bordering the montane, so it has a bunch of different plant species like Festuca campestris, and it also has a lot higher temperatures. So I think this is going to inhibit our ability to directly compare the veg communities in fire versus harvest, but we can certainly still compare the microclimates by using relative temperatures rather than raw temperature values, but not really much we can do about those unique vegetation communities. So moving on, here's some data showing our microclimate as we move across the edge distance. So on the left of our x-axis, that is the interior of the disturbance. On the right of the x-axis is the interior of the remnant. So you can see that as we move from the interior of the disturbance to the interior of the remnant, our temperature ranges get smaller. So the difference between maximum and minimum gets smaller. And then also decrease. So typically we have higher temperatures in the disturbance relative to the interior. We also have a peak at about -15 for the maximum air temperature, which is kind of interesting. This might have something to do with the amount of airflow around the forest edge. Maybe that those additional trees or snags that are along the edge are preventing mixing of air and may kind of create a stagnant effect that allows that area to heat up more. So here we can compare the air, surface, and soil temperatures between fire and harvest on the left. We see that both disturbance types show a similar trend of decreasing temperatures with increasing edge distance. And the same here is seen on the right when we compare our islands versus our reference sites. So those islands, those are going to be our little patches of trees inside the disturbance, whereas the reference is going to be a large intact patch of forest adjacent to that disturbance. So we can see that these islands show a similar trend to the reference, and that also the difference between 45 meters and 100 meters is not too great, which supports the idea that these islands can have similar microclimate conditions to the interiors of larger forest tracts. So here we looked at plant community response to edge effects. Here I did remove those outliers. So this is just looking at harvest and a couple of fire sites. And yeah, we can see that there is some variation here. The disturbance is shown in purple and the interior is shown in blue. As expected, the edge kind of straddles those two spots. The black temperature vectors, they do indicate a greater range in temperature inside the disturbance compared to the interior, as we just saw. But we're not seeing any, super obvious effects of vegetation, maybe Vaccinia myrtleoides there, associating with the high temperatures of the disturbance. Maybe Camerion angustifolium also associating with disturbance, and Salix pseudomyrsinites associating with interior. And then we can see some of those veg vectors associating with each other. Again, these are just the top 15 or so vectors as far as their influence, so there might be others that we might see if we look at the other 180. All right, and then I looked at the differences or the similarity in vegetation communities between islands and reference sites, and these looked pretty similar. I didn't see any obvious trends in terms of temperature associations with either one or with certain plant species. It did seem like the island edge, which is the pink dashed line, was the most different from the island interior. But reference edges and reference interiors were pretty similar. All right, so the next kind of things I'll have to do with this project are to look at plant functional traits. We might consider other ways to deal with that outlier site, but obviously we'll run our statistics and see what significant effects we can find.